Ladies and gentlemen, good morning. Welcome to the second day of the INCOSE conference. Our first point on the agenda is the keynote by Hendrik Hofmeyr. If you study systems thinking or systems approach or anything about systems, which is the basis of systems engineering, you will discover that engineers didn't discover these wonderful principles. It really originated in biology, in the life sciences. Concepts such as the hierarchy, emergent properties, ecology, food change, that's all from biology. It is worth our while, every now and then, to calibrate ourselves as to what biology is doing about these principles and whether new principles are emerging. Jan Hofmeyr will present us a keynote entitled The Complexity of Life by Hendrik Hofmeyr. Hendrik Hofmeyr is a distinguished professor of biocomplexity and biochemistry at the University of Stellenbosch. He is a co-director of the Stellenbosch Center for Complex Systems in Transition. Why is he distinguished? Well, quite apart from academic issues, he is the national chair of the Arthritis Foundation. He received the Hofmeyr Prize for the biological systems a few years ago. He's a musician, the flute and the guitar, and is also a cabaret artist and actor who helped launch Afrikaans cabaret during the 1970s and 80s. He also climbs mountains, Kilimanjaro and the Himalayas. He's a regular participant in Hoe verklaar jy dit? On radio Sonder Mense, a question and answer session on science. In the early 1970s, he did his MSc on metabolism, using differential equations as a model. But he discovered that you cannot explain metabolism in isolation from its environment, which of course is the human body. He discovered that this biochemistry is very reductionist. The word was also used yesterday, where you have a system and you try to explain everything of the system in terms of its components. However, what counts more than the environment, in the components rather, are the interaction between those components that create the so-called emergent characteristics at the level of a system. Jannie just hence joined the systems biology movement, which propagated that. Investigating a system in its environment is great, but it doesn't help you. Explaining what those emergent properties are, do they just arrive by accident? The fact that you can measure it doesn't mean to say that you understand how it works. So Hendrik Hofmeyr teamed up with the late Paul Sillier to start working on complexity theory. Hence, he will now present a keynote, The Complexity of Life. Thank you, Jannie.
There we go. I don't even have to stand in front of this box to say good morning to you. Thank you for coming. I'm very happy to be here. And I want to thank Ad for the idea of inviting me to come and speak to systems engineers. And I must say, I find it very exciting because for the first time I have an audience that I think is going to understand what I say. Because it's all about systems. It's all about how things fit together in order to make something work. And how can we design something that does a very, very special thing. And I'm going to talk about that particular thing. There we go. So recently, there was a Facebook message by a colleague of mine said, I wish I could start a paper with such a nice sentence. And this was a paper that I wrote together with a colleague of mine, Olaf Volkenhauer from Rostock. And I immediately said, well, I didn't write that sentence, but it's very nice, I agree. If an organism is our universe of discourse, the cell is the star we gaze at. In its complexity and functionality, even the simplest, tiniest cell dwarfs everything humankind has ever been able to engineer. And all those red words are the words that are going to be important, especially the engineering part. I would have said fabrication, that is the word I prefer to use, but in this audience, engineering works well. My colleague said, well, he didn't write it either. He must have got it from somewhere, but he can't remember where. So anyway, I thought that is in any way a nice sentence to start with. This is an amoeba. It's a little piece of soft matter that does something that we call living. We immediately recognize it as living. We don't know why, but it's an intuitive thing. The question is, can we understand what makes that piece of soft matter living and other matter that we know non-living? So what is the difference between living and non-living? And in a sense, that's what I want to talk about today. So let us ask ourselves, what are the most fundamental challenges any organism, from an amoeba right up to you sitting there, what are the challenges we face? Well, the first challenge we face is we have to distinguish ourselves from the environment in some way. We have to defend a little piece of space in the universe so that the rest doesn't encroach on us. Then comes a very important point, is that all living systems are built from fragile components. In a year's time, there will be virtually no atom left in your body that you have in your body now. Yet your mom is still you're going to be slightly worse for wear, but your mom is still going to recognize you, you're still going to be you. So in a sense, what you've been doing is you've been living longer than the lifetimes of all the components inside your body. That's an amazing thing if you think about it. And that's what we're going to talk about today. So you're built from fragile components. Any organism has to, there are environmental fluctuations during its lifetime, and it has to be able to adapt to that. And then you have to cope with environmental changes over generational timescales, and this is something that, for instance, Pierre talked about yesterday, the whole idea of evolution and how it works. That's not what I'm on about. I am particularly interested in the second one. So there we have various forms of self-bounding, that a cell has a membrane around it that is semi-permeable. It allows certain things to come in, pumps certain things in, pumps certain things out. So it bounds itself and so it distinguishes itself from the environment. There must be some other functional organization within a living system that allows it to make itself in order to overcome this problem of being built from fragile components. There we have mechanisms in any organism, from a bacterium up to us, that senses the environment and allows the system to restructure itself, adaptively restructure itself within genomic constraints. Of course, we have a genome. The genome determines what we can and what we cannot do, what we can and what we cannot make. Beyond that, we have to change the genome in order to cope with these evolutionary adaptations through descent with modification. So this is a whole year's course in order to talk about this. I only want to talk today about that particular aspect because I think it's the aspect that's going to be interesting for you as engineers. How do we make a system that can build itself? How can we build a factory that can manufacture itself completely, autonomously from within? Right, so that is the project for this morning. So this self-fabrication is, if Pierre yesterday talked about various properties that you can confer from living systems or infer from living systems, I would say self-fabrication is the most fundamental of all, because all these other properties that we usually associate with life cannot happen if you cannot make yourself to start off with. And I'm going to, so growth, reproduction, evolution, maintenance, adaptation, metabolism, they all depend on this aspect of this property of being able to make yourself. So let's take one example. Growth comes before reproduction. So what happens? If you want to grow, say you're a bacterial cell, and you want to reproduce, there's the cell. It has to grow to twice its size and then divide in order to become two cells. So what does that growth entail? It's not this sort of growth. It's not growth by expansion, although there is expansion, but it's not just increasing the volume. It's not growth by accretion, like a crystal, where you have a particular type of compound, let's say sodium chloride, and you have a seed crystal, and then dissolved sodium and chloride ions accrete onto a crystal to make it grow. It's not the same than that either. It is something completely different. It's growth by self-fabrication. And the difference is the following. If you want to compare, for instance, accretion with self-fabrication, there you can see you have a seed in the middle and the same molecules just accrete onto the crystal to make it grow. But growth by self-fabrication means that you have a system, it takes in food, let's call it food, from the outside, it transforms that into itself, and in that sense, it can become bigger. So all the molecules, and the huge diversity of molecules, actually duplicate themselves inside the cell in some way. So that's the difference, for instance, between a car that takes, you put petrol into the car burns the petrol for energy, but it doesn't, that's all it does. It just gets energy from the food. You do something different, you eat food, and it becomes you. You transform that food into you. Of course, some of it is used for energy, but some of it is used for building blocks inside the cell. So that's the difference. There's also, if you can think, there are two ways of thinking about self-fabrication. One is to grow to twice the size and then divide, but the other is just maintaining yourself all the time. And that's what you're doing as you're sitting there. The breakfast that you eat is now becoming you, and you are making yourself as you sit there. And every living organism does that. So renewal by self-fabrication would need the same sort of functional organization in order to do that. Right, so that is an introduction. This is what we want to talk about, self-fabrication. From an engineering point of view, let's put it that way. For that, we need two types of thinking. I'd refer to the fact that reductionism was mentioned yesterday. Of course, it is extremely important that we are able to do that. I call this machine-age thinking. It's important that we analyze things, that we understand the structure of things, how things work, because that yields knowledge. It gives us facts that we can use in our thinking. What we do when we do machine-age thinking is we describe a system. We look from the outside into things. So I'm going to use an example that virtually everybody knows, those that like music especially, a hi-fi system. So there's an amplifier. Machine-age thinking, if I give you an amplifier, what do? Screwdriver, all right? And you open it up, and how does this work? What does this thing do? What does that thing do? So you analyze it. Circuit board, and you go into the circuit board, you say, well, that little thing that, what does that do? And what does that do? So analyze. Have to do that. Otherwise, we don't know what's going on inside that system. Of course, we try to reverse engineer as well, but this is an epitome of what we do in machine-age thinking. But that's not enough. We also need to go in the opposite direction. We need to do systems thinking. So instead of analyze, we have to synthesize. Instead of looking at structure, we have to think of that particular thing. What does it do within the system? What is its function? Not how things work, but why do they work? That's going to be an important word, that why. Why does something work? Can we explain it? Yields knowledge on the one hand, yields understanding on the other hand. Of course, that's what, as systems thinkers, we are after that deep understanding of a system. Not only describe, but explain. Look into things, yes, but on the other hand, we have to say there's a component. It must be part of a larger system. What does it do within that system? This is, I hope, something that you all, of course, do in any way. Yeah? As systems thinkers, this is what we have to do. But sometimes we hear, our reductionism, we have to get rid of that. We can't. That's just as important, going up the ladder and down the ladder. So, amplifier. Of course, if we do systems thinking, we think, okay, the amplifier it must have a function inside a larger system in the hi-fi. So, of course, there must be some input to the amplifier, and then some input signals that get amplified and then get, for instance, to a speaker. So, as soon as we do that, and we think of how things are coupled to each other, and the relationships between things, we need to think about organization. And how can we understand and describe organization within systems, in general, not only in life, but in any system. And this leads us to something which has become very important. It's called relational thinking, which is a part of complexity thinking or systems thinking. So in the reductionistic approach, we are interested in the structure. and how things are fit together. But if we look at relational thinking, we throw away the organization when we do that, when we analyze. We break things up and we break up the organization. Relational approach, what we do there is forget about the matter and just abstractly think about how things are organized. And we have to have a language to do that. And that's very important because we don't have formal languages. We need a formal language in order to describe organization. And I'm going to take a little bit of time to show you such a language, which has been extremely useful, especially when we think about living systems. So the concept of function of a component within a system is of crucial importance. It's the heart of our intuitive use of organization. Organization is everything to do with function. So what I'm going to tell you about now is the brainchild of a theoretical biologist who lived in the latter part of the previous century. He's called Robert Rosen. Again, one of these people who were too far before their times. And nobody really listened to him while he was saying very important things. But he had this question, what is life? Which drove his whole research career, from young PhD student right till the day he died. And in that journey, he developed a way of thinking about organization. So he says, one must think on any organization of how do you describe a component within that organization? He calls it a particle of function or an atom of organization. So very abstractly, if you have a system and you delineate a component in it, then there's always going to be input from the system and from the environment into the component, and output from the component to the system and the environment. And his genius was that he was able to capture that in a mathematical mapping. So he lived at the time just when he lived in a time when there was a new form of mathematics called category theory. And he was fortunate enough to be at the University of Chicago at the time when Samuel Eilenberg and Saunders MacLean, the two developers of category theory, were actually teaching there. And so he was the first one to go out of very, very abstract mathematics and realize that this is a way of talking about organization. Classical mathematics based on sets, as you know. Category theory is based on the idea of mappings, of morphisms, of arrows between objects. And it's those arrows, the processes, that are the most important part of category theory. So it's a perfect language to talk about processes. So that's a simple mathematical mapping. You say the component F maps inputs A into outputs B. And what he did, and this was actually, it looks so simple, but he put that mapping into the form of a graph in which the function, the component, is actually a node in the graph. It's not something that sits outside the graph, it's something inside the graph. In other words, it's something that can be the input to something else or the output of something else. So you can actually use these mappings then to build up a picture of how processes are organized. So that's this, and I'm going to use this a lot. So the dotted arrow is, we're going to show the mapping, the thing, the operator, the agent that acts on inputs to give you outputs. So, amplifier again, just as an example. So there's an input signal and an output signal. And so we can write that particular component, amplifier, which acts on an input signal to give us an output signal in the form of this graph theoretical mapping. And of course, we don't only have to think of the amplifier as the component, we can think of the source of the signal as a component and the receiver of the amplified signal as a component. So this is a system containing 3 components. Each of those will be described by such a mapping. So you can think of putting the organization, which is a linear organization in this particular example. You can say that in very general, there's a set of audio sources. The receiver acts on that to give you a set of input signals, output signals, sounds, and then you can choose a particular example. It's called chasing elements through the sets. So you can say, okay, my audio source is a CD player, gives me a signal, digital signal. Then there's a converter, that doesn't matter. Converter, and then amplification, and then music in the end. And of course, the nice thing is with these mappings, in category theory, you can compose the mappings, which means you can join them together into a whole. So here is you can say there are three processes work together to give you in the end a hi-fi. So whether you talk about the components of the system or whether you talk about the whole system, it allows you to understand how the system is put together. In this case, a very simple one, a linear set of mappings. So this would be abstractly the way that we put those three mappings together. You can compose them into one particular mapping. So this is the mapping of the whole system. That's the mapping that shows how the components fit together. So that is the formal language that we can use. And we said what we want to do is to understand the system. So we want to answer the question why. Why that system? Can we explain it? And now we go back to old Aristotle, who thought about this very carefully. You may have heard of Aristotelian causes, material, efficient, formal, final causes, somewhere in your life. They're not causes, they are becauses. They explain things, 4 ways of explaining something. So I'm just quickly going to go through this because I need the terminology. So if we ask for any artifact, say artifact X, it's useful to say, what is X made out of? What makes X in the sense of what produced it? What makes X in the sense of what is the essence, what it is to be X? And finally, what is X made for? So it's very useful to use this verb make. And of course, for us, engineers, this is very important. We do make things. And this is a way of explaining it. And these, if you can answer all of those questions, Aristotle said, you've completely explained that artifact. That's called the material cause, or because, the efficient because, the formal because, and the final because. Nobody else calls them becauses, but I do because they are really answers to questions, the why questions. So in terms of the amplifier, what makes the amplifier, who makes the amplifier, what makes it in the sense of what is it to be an amplifier, the essence of it, and what's it made for, what's its purpose or function. And of course we know, material cause is all that stuff that we see when we open the box, all the little bits and the transistors and the capacitors and whatever, and the circuit board. The efficient cause of an amplifier is the electronic engineer that built it. The formal cause, of course, is the plan, the blueprint for that particular amplifier. And the final cause, very simple, is to amplify a signal. So in terms of the things that we make, it's always, not always easy, but it's possible to give a complete description of such systems in terms of the Aristotelian becauses. For us, the most important part, and you'll be glad to know that, is the engineer, the efficient cause. Because what we in the end will ask ourselves is, how are the efficient causes in living systems produced, and how do they produce each other? So again, I'm going to focus in the end on that one. And another stroke of genius of Rosen was that he realized that his mapping, this graph theoretical mapping that he produced, actually mapped the Aristotelian causes. It's very simple. So F is always the efficient and the formal cause working together. It's the engineer plus the information that then acts on the material, in this case all the components of the amplifier, say, to give us the effect. The effect in this case is the amplifier. And so I'm not going to talk about final cause now because it's possible to bring it in here, but it just gets confusing. But in effect, the effect is the final cause of the efficient, formal, and material causes. But then it becomes a bit complicated. I don't need all of that superstructure. But this is important, that he was able to use his mapping to talk about causality within the system, how things fit together in terms of what is an efficient cause, what's a material cause, and so on. So, let's build a factory. And I want to build a very special factory in the end. I want to build a factory that can make itself. Well, we haven't been able to do that yet, but at least we now have a language to talk about how we would possibly go about it and what the possible problems may be when we do this. So think about, in very abstract terms, simple factories as we know them, or build them. You have raw materials, input, there's a series of machines on a conveyor belt or so that produce from the raw materials some other output, some other product, that is the product of that particular factory. The machines are the efficient causes. I can write this, of course, in terms of such a mapping, but now I'm just going to build up a slightly different picture. But keep in your mind, efficient cause, material cause, effect, and the end. So, that particular factory I can then write as a sequence of material transformations, from raw materials through intermediate products to the end product, as a series of material causations. So a linear path of material causation. You see, it's very useful to have these words now suddenly and a picture of how these things fit together. Of course, it doesn't have to be a linear path. You can always have cyclic paths of material transformations. We have this in our bodies. Every cell we have in metabolism, we have these cycles. The so-called Krebs cycle or citrate cycle is one of them. That's a cycle of material transformation. But you can see the dotted arrows sit outside the system. They're not produced from inside. They're given from the outside, just as they are at the top. So that's a closed path of material causation. Now we have a problem. Those machines, of course, have a limited lifetime. They will at some stage break down or have to be repaired or replaced or whatever. And the question is, when they fail and they have to be repaired or replaced, who does it? And we know in our factories it's some other engineer that has to come in, or a technician that has to, or the guy in the office that has to buy a new machine and complains about it all the time. So there is a higher level of efficient causation that is necessary. But we can think of, so this is a way of putting it. So if you have all of these machines, F, G, and H, for instance, you need perhaps, in this case, humans A, alpha, beta, and gamma, that come in and repair those machines. But we can think of a situation where we have a more complicated factory. Sorry, I just need to go past. We have a more complicated factory where that second level of efficient causation are not humans, but other machines. Machines that repair machines. Yeah? That's possible. Why not? So, instead of the humans, we have now a second level of machines that repair the other machines. But of course, you can see what's coming. What happens if they fail? Then again, you can build perhaps those machines from products inside the factory, but you still need humans as a third level now in order to repair the machines that repair machines. And you can see we have a problem here. There's going to be, in the end, an infinite regress if we carry on like that. In the end, there's always going to be an upper level of efficient causation that is not produced from within the system. So the question is there a way in which those machines, in a sense, perhaps can make themselves, that can use endogenous information within the system to build themselves? Or is there some way of closing that loop, that efficient causation loop? And of course, as I alluded to right in the beginning, you're doing it all the time. So you are examples of systems close to efficient causation. But how is that possible? I mean, if you look at it this way, it's not immediately clear that this is at all even theoretically possible, that machines can actually make themselves. So what do we have here? We have such a hierarchy, and the hierarchy closes in on itself. So in a sense, those two black dots become superimposed, and you form cycles, either a two-member or a three-member cycle, no matter, depending on the side of the hierarchy. But the crucial difference from what we had before is that the arrows are now part of the cycle. The arrows are produced from within the cycle. They produce each other. And this is called the hierarchical cycle. So there we have a hierarchy of processes in terms of functions producing functions producing functions. Now here we have a system that's, where the functionality has closed in on itself, and the functions produced each other. This is already known philosophically from a long time. Immanuel Kant, for instance, one of the guys that really formulated this nicely. He said everything within a living system is there in terms of something else in the living system. They all produced for each other. But that's where it stayed. There's also, you may have heard of the term autopoiesis. This was thought of by two Chilean scientists, Humberto Maturana and Francisco Varela in the 60s. They also had the idea of self-production. Autopoiesis means self-building. But they never did anything with it further to explain how it really works within the cell. But Rosen had this formal language, and that's what I like. It gives me a grip on things. So now I want to try and understand how this works. And what is the problem with this sort of system? The problem is that it forms a closed path of efficient causation, so that F causes G and G causes F, yes? But that is a self-referential cycle, and it always has, as soon as you have self-reference, you have a paradox. Because things are defined in terms, in the end, and circularly defined in terms of themselves. And that's logically a problem, often. But we have to get past this. So impredicativity is an important part of the whole story. I've got a picture here, but I'm going to skip this. This just explains in the end how you can put all of these things together. I just want to point out that Y, which is a set here, does this thing have that Y, can you see, is defined in terms of itself. I'm not trying to explain all these things, but Y is this function, and Y occurs inside it. So Y is defined in terms of itself. That's a problem. But it's a problem that can be overcome. It's not, even in mathematics, impredicativity is not always a problem. So this is the difference between a cycle of material causation and a closed cycle of efficient causation, the fact that the arrows are inside this. And Rosen realized that this is a very nice way of distinguishing complex systems from non-complex or simple systems. So this is the basis then of what is now called Rosenian complexity. He said that a complex system will always contain one such cycle. Not all the efficient, what did I do now? Not all efficient causes have to be inside the cycle. There's one that sits outside, but at least there must be one such cycle, self-referential cycle, within a complex system. And if all the efficient causes, like here, are inside the system, produced within the system, then you have an organism, a living organism. We are the only completely closed systems of efficient causation. So this is very important. So Rosen doesn't talk about things become more complex and complex. He says, no, there's a divide between simple systems and complex systems. Complex systems always have such a self-referential cycle in them. In Cape Town, there's a thing that happens every 12 o'clock in the afternoon, and that is the noonday gun. So boom, from Signal Hill on 12 o'clock. That's part of Cape Town life. So there was a tourist, and he had heard about the Signal Hill noonday gun, and he thought this is something he has to investigate. So he went up to where the guy who shoots the gun, and he said, listen, this is amazing. How do you know when it's 12 o'clock? Because your watch may be wrong, and so on, and maybe you're off by a few minutes. You say, no, I'm always exactly right, because down in Long Street, and with my telescope, I can see the front of the guy's shop. There's a watchmaker, a famous watchmaker. And he sets his clocks perfectly. So there's a big clock in the window, so all I do is I look through my telescope, and then I have my finger on the trigger here, and if it's 12 o'clock, I go boom. So this guy, I have to meet this watchmaker. So he moseys down to Long Street, into the shop, and he introduces himself, and he said, I've heard that you are such a fantastic clockmaker. And your watches are always correct. How do you set your watches? He says, very simple. There's a guy up there in Signal Hill, 12 o'clock. That's a perfect example of an impredicative system, defined in terms of each other. And actually, think about it, it'll work. For them, it'll work. They'll never know that they're out of sync. It may be that they drift and that they are not at all in sync with atomic time for the rest of the world. But within that system, it functions, right? And of course, sometimes it needs an adjustment from outside. Somebody says, listen, guys, it's fine, you two with your internal system of timekeeping, but you need to adjust a little bit. But the point is that impredicative systems can work. And you, again, I say that you are examples of that. I'm not going to, so in the interest of time, I'm not going to talk about this, but again, there are various ways of describing organizations. Russ Ackoff did it, and his way of explaining it maps nicely with, you can describe it in terms of Rosen as well. What I want to spend the last part of my lecture on is, now I become a biochemist again. And I said, listen, I understand most of what happens inside the cell, but nobody's ever tried to see how everything fits together in order to make it self-fabricating. Rosen had done that, but there was a problem. So he built for himself, remember he asked himself what is life, and he tried to understand how everything fits together with his mappings and his Aristotelian causalities. And he said, well, at least at the heart of all living systems, all organisms, there is what he called the metabolism repair system. A is the food that you eat, and that gets transformed by a whole complicated machinery of enzymes that catalyze all the reactions inside the cell, the metabolic reactions. It gets transformed into building blocks. The building blocks for the macromolecules that we have in our bodies, in our cells. Proteins, complex lipids, nucleic acids, and complex sugars. But they all form strings. So life is basically based on string building, on polymerization reactions. But then he said, what's the problem? We need to explain where F comes from. So he said, well, it's easy. We can just think of another mapping that uses the building blocks, he called it the repair mapping, to build the machinery, the F machinery, the metabolic enzyme machinery. But then now we have this phi function here, which sits outside. Can you see? We've pulled one efficient cause into the system. That's now produced within the system. But now again, we have one that sits outside. So he asked, what repairs the repair system? And of course, you can think of another mapping. You can use F in order to then produce phi, but then you sit with a beta mapping, which sits outside the system. Can you see, again, this idea of a sort of an infinite regress. If I now ask what's beta, I have to think of another efficient cause. And his genius, again, as a mathematician, he was able to mathematically at least pull the beta into the system, so that he didn't need to have a map that sits outside the system. And he called that a replication map, which is a very unfortunate term because it's got nothing to do with replication. In fact, it has nothing to do with anything. It's a cute mathematical way of pulling that map into the system, but he couldn't explain what it meant in terms of biochemistry, in terms of how we know the cell works. And this is how I became really interested, because I was very unhappy with that. I said, no, I cannot go around talking about this if I cannot explain what that orange mapping is. I have to know what it is. There must be something wrong with the way you put things together. But the idea is that you have three mappings which superimpose onto each other, and if you chase elements through these mappings, you have a cycle. There's a clear hierarchical cycle. So his little system that he has is one that's close to efficient causation. It's a hierarchical cycle, fulfills all the criteria, except it doesn't map onto what we know what happens inside the cell. And so I want to take you on a journey through the biochemistry in 20 minutes, what I usually teach in 3 years. But you'll see, it works. I think I've figured out a way how to do this. The first thing is that, it's a bit okay. If you walk into a biochemistry lab or a biochemistry lecture hall, you'll see such a map on the wall. This is all the metabolic reactions that take place inside a cell. And this is the metabolic machinery that does it. There are lots and lots and lots of complicated molecular machines that do various things, that catalyze reactions, that break things down, that build things up, that give information, and so on and so on. We're going to forget about all of that detail, and we're going to condense this whole map and all the machinery into a very simple picture. So again, this can be thought of functionally in a very simple way. You get nutrients in. There's a whole set of metabolism we call catabolism that breaks the nutrients down and gives us energy and reducing power. Energy in the form of ATP, adenosine triphosphate, and NADP gives you reducing power. So without that, you can't get the process to run. You need energy, chemical energy. And it gives you carbon skeletons that are the building blocks for further synthesis, which we call anabolism, and they give us the building blocks, amino acids, nucleotides, fatty acids, that we can use to build up biopolymers, proteins, nucleic acids, and complex lipids. So this is the same map that we had before, except now we look at it in a functional way, functional organization. This may still be too complicated for us, so all I'm going to do is that. We're going to work with one map, catalyzed covalent chemistry. So the chemistry that underlies all of those hundreds and hundreds of transformations is all the making of bonds or the breaking of bonds, covalent bonds. For those of maybe if some of you have done chemistry, but that's a particular very stable sort of bond that give us structure. Our atoms are linked together. And all of those reactions are catalyzed, each reaction in the cell, thousands of reactions in the cell, are catalyzed by a particular enzyme, specific for that step, specific for that particular substrate on which it acts. That's where the catalysis comes in. So I've now made one map. This is one map inside the cell, one efficient cause. That's the F going from A to B in Rosen's story, right? So that matches on what he said, except he didn't use this sort of terminology. So the efficient cause 1, the first efficient cause. Remember, I'm now looking for the efficient causes and see how they fit together. That's, you can call it structural entailment. It's the building of structure. If you think of a human organization or a society, you can now think of the people that constitute that particular organization or that particular society. Let's say the raw people. They're not functional yet, but at least they are there. They've been co-opted from outside and trained to a particular point, and they are about to do something, but they're not yet functional. In the cell, let's call it the covalent chemistry of intermediate metabolism, that's catalyzed by enzymes, and then more complicated machines called ribosomes that are specifically for protein synthesis. And then also you have in the membrane of the cell, you have proteins that transport the food into the cell, the nutrients into the cell, they're called transporters. So those are the first set of efficient causes. So I'm going to build up a sort of an arrow picture for you to see how everything fits together. So here's this one map that I've showed you, intermediate metabolism, from nutrients here to building blocks. And this is all catalyzed by enzymes. And amino acids, nucleotides, and fatty acids, those are the building blocks. Those building blocks are then used to make very important compounds. Obviously, DNA, various forms of another polynucleotide called RNA. Remember, Pierre talked about RNA yesterday. There are 3 major forms of RNA. The green one, ribosomal RNA, is a part of those ribosomes that I talked about. Messenger RNA, you'll see the role of it now, and transfer RNA has to do with the genetic code. How do you translate one molecular language to another molecular language? Something I mentioned yesterday. So amino acids go to polypeptides, nucleotides to DNA, RNA, fatty acids to lipids. Lipids are, of course, important. They are the constituents of the membrane around the cell. All of those reactions are catalyzed firstly by enzymes, and that's what the blue dotted arrows are. Remember, the dotted arrows are efficient causes, right? The solid arrows are the structural transformations. So DNA, and the question is, how do the enzymes know how to make these polypeptides? Well, the information for making it comes from the DNA. The DNA is translated into an mRNA form, and at the end that's translated into protein sequence. The second part, so you see here what we've left. We left here with amino SRT RNAs here. Forget about the fancy name, but they are just activated amino acids. They have to now be transformed into polypeptides, and that's done by the ribosome. So there's another efficient part of the efficient causes, another dotted arrow. And that information then comes from messenger RNA. So the genetics part of it is how the information flows. Those are the small dotted arrows from DNA into messenger RNA into proteins, in the end into polypeptides. And the nutrient transporters, so now I've drawn a cell membrane around it, and in the cell membrane there sits a transporter that transports the nutrients into the cell. So those are three parts of the first set of efficient causes inside the cell. Now the important bit here is that the polypeptides are not yet functional. Just as I said just now with the human organization, you can train people up to a particular point where they have the skill, but they're perhaps not yet functional. What do they need to be functional? They need to be in the right environment. If you have an organization that has a toxic environment where nobody can work, then no matter how skilled your people are, they won't be able to function. The same with these polypeptides. They need a very particular environment in order to become functional. And in order to become functional, so a polypeptide you must think of as just a long string of amino acids coupled together. Before they become functional, they have to undergo certain processes. They have to fold up into a three-dimensional structure in which there's a particular architecture called the active site, which recognizes the substrate and then can convert it catalytically to a product. The enzyme itself is not changed in the full process. That's why it's a true catalyst. Sometimes even these folded proteins clump together by self-assembly into higher order structures like the ribosome, for instance. And in order for that to happen, we need, we need a folding and assembly, we need an intracellular milieu. There must be an environment within the cell which allow those processes. They're called supramolecular processes. We've had covalent, now we have the folding up, the making of three-dimensional structures. It's a whole new field in chemistry these days. There's already been one Nobel Prize for it, supramolecular chemistry. And for that, we need the right environment. It's called the intracellular milieu, and there are even proteins called chaperones that help the folding process to prevent misfolding of the proteins. So a chaperone is like a microenvironment in which the folding can take place. It can happen without the chaperone, but the chaperone just makes sure that it's absolutely correct. So this is where we were just now. Polypeptides must become functional, and in order to do that, the polypeptides have to, all the orange arrows now, or the yellow arrows, polypeptides must go to enzymes, they must go to ribosomal proteins, they must go to chaperones, and the lipids must self-assemble into the membrane. This all has to happen due to the intracellular milieu. So the folding, the self-assembly, and the self-assembly of the lipids all is catalyzed. And really, one can think of it as catalyzed by the intracellular milieu. So this is again something that is new here. Nobody's ever thought of the intracellular environment as an active agent. Just as nobody seems to think of the environment within an organization as an active agent. It's not something that people think of really creating actively every day. So there's a lesson from the cell here in terms of our own organization, our own societies. We have to have an environment in which one can be functional. Think of the South African situation at the moment, and we've fallen into a situation where many people are, they're perfectly capable of doing what they want to do, but they're not in the right environment. And so the cell has to actively create this environment, and this is what I think is new to the story. So, the intracellular milieu and the chaperones, all the dotted arrows again show they are an efficient cause within the system. But now the question is, of course, that particular intracellular environment or milieu has to be created from within. Otherwise, there's again an efficient cause that sits on the outside that checks that everything is right inside. So how does that happen? And so this is where the transporters for electrolytes comes in. So what the difference is, so the transporters are proteins that sit in the membrane, and they transport specific ions. And can have the sodium-potassium pump, the calcium pump, and so on. What they do is they maintain an intracellular environment that is completely different in terms of electrolyte composition, ionic strength, pH, protein concentration, than what you find on the outside of the cell. Here's a little table. If you compare, for instance, the cytosol, which is the fluid inside cells, together with, for instance, blood or with seawater, you can see it's very high in potassium, in contrast to very low values outside. It's very low in sodium compared to very high values outside. Chloride, it's a big difference. Bicarbonate, not such a big difference, but quite substantial, especially between cytosol and blood. Lots of amino acids and proteins, and they act as buffers, pH buffers, you don't have outside the cell. Calcium Calcium is a particularly important compound, and it has to be very low inside the cell. High calcium concentrations cause a lot of problems. So you can see what these transporters do is they maintain an intracellular milieu that is conducive to function, and very, very stringent homeostatic ranges in which these can vary. So it's a very constant environment that is kept from within, not from the outside, but from within. So this is the picture of that. You need cations, anions, and they are particular composition. You need buffers, phosphates, and proteins. And this is maintained by the... I always press the wrong button. It's maintained by the transporters. So we have closure to cell fabrication. We can show what the efficient causes are, and we can show how they fit together. So we have the first set of efficient causes, molecular machines that catalyze covalent chemistry. We have the second set. This is the intracellular milieu that catalyzes the supramolecular chemistry that makes things functional. And we have the electrolyte transport that maintains the intracellular milieu. And that is how the whole cell manages to make itself all the time from within, autonomously. This is when you put everything together. It's a very complicated picture. This is a slightly simpler picture. You can see there's all the metabolic machinery that we need. This is how it's made. This is the supramolecular chemistry, and that is the way that the supramolecular chemistry is maintained. So again, another picture of how everything fits together inside the cell. Let's call it the chemical factory that can make itself. And I could even now make my own little abstract model. So there is the first set of mappings, nutrients going to polypeptides, catalyzed by enzymes and ribosomes, and then you have the intracellular milieu that makes those polypeptides functional, the yellow arrows, and then I have the maintenance of the intracellular milieu by electrolyte transport, and they are already there. These are just inclusion arrows to show that C is already there, and T is already there. So this is another way of writing that mapping, the set of mappings that close each other up. There are the three mappings, and if I chase elements through it, then I also have a hierarchical cycle. But now each of my mappings map exactly onto a process that we know inside the cell. So this is similar to Rosen's mapping, except that I think it matches on what we know. And that to me was particularly satisfying to be able to make that picture. So I want to end with two slides. The one is, there's a very famous mantra for evolution in biology. It says, nothing in biology makes sense except in the light of evolution. But that sort of picture, you know, if you ask an evolutionary biologist, why do I live? He says, well, because your parents lived. Why did they live? Well, because their parents lived. Yeah? Which is a sort of an answer, but it's extremely unsatisfying for somebody who wants to understand why does that particular organism live, right? So a mule, for instance, cannot reproduce, yet it's living. It has this organization that allows it to self-fabricate. So I've got a mantra that I have for systems biology. Nothing in an organism makes sense except in the light of functional context. And I think that is the crucial point, that if you think of systems, you have to think of context, always. What makes something functional? What is the context that allows things to happen for every component in the system? And so that mantra actually, as I've been trying to sort of interject in between tells us something about what we can learn from the cell, about our own organizations, and about our own societies, and what is necessary for making a persistent organization or a persistent society. Because if you think about it, in organizations, components are also fleeting. They're also fragile in a sense. They come and they go, yet you want the organization to remain. So you have flow of material through the organization, yet you want the organization to remain as an emergent property, yeah, of all of those interactions. So again, this particular idea of emergent properties in organizations is extremely important. And the trick is to really consider the environment of your organization or the culture of your society, because that's basically the milieu for a society is its culture, to make sure that you actively maintain it. Because if you lose that, you lose the organization and the system breaks up in the end. And with that, thank you very much for your attention, and I hope that we can have some chats about this today still. I'll be here. Thank you.
Ladies and gentlemen, that was wonderful. Are there any questions?
Okay, so that process you described, it's quite sophisticated. So I was wondering if you think of how a human body gets developed from embryo stage to final. How does that structure actually get built from just a basic fetus, or no, I mean, what manages that basically?
You're asking a jackpot question here. So this is, embryology, of course, is if you think it, utterly amazing. You have a single cell that grows into you, yeah, where you have thousands of cells, over 200 types of cells. But think about it. You begin with this organization, this self-fabricating organization, and a genome. So in the beginning, not everything is expressed. And so as you start expressing through this process different parts of the information that is in the genome, in a very controlled way, and we're still not quite sure how all of that works. So embryology is still, on a molecular level, is something that we cannot always explain. But in some way, that controlled process unfolds what's inside the genome, makes, under certain conditions, certain parts of the genome are expressed to form differentiated cells, so a muscle cell, or a red blood cell, or a liver cell. And slowly, in the unfolding of this genomic information, you have the creation of a multicellular organism with differentiated functions. More than that, it is difficult to say here now. Of course, it's a hugely complicated process, and we understand a lot of it. But basically, you have the information in your genome. It just needs to be, in a very controlled way, unfolded through stepwise division of cells and making more cells and more cells, and then starting to differentiate them and sending them to different parts of the body. So very interesting to follow the embryological process to see what happens first, and the neural crest forming, and then suddenly you have a top and a bottom, and the cells orientate themselves. There's gradients of molecules throughout the system that control it. So it's a very complicated process, and for me still, when I think about it, just utterly miraculous.
I thought there were no miracles.
Yeah, miracles, not real miracles, but I mean, you have to have a sense of wonder. So if you're a scientist and you lose your sense of wonder, go home.
I'd like to take you back to your introduction on the systems thinking part. And if I look at this, I'm struck by the level of certainty with which you speak. And for me, I'm experiencing quite an amount of cognitive dissonance when I work with complex systems, because I experience a lot of uncertainty. And one of the things that I'm also reflecting on a lot is, there's this sort of almost domineering attempt to define system. Who will conquer the definition of system, and that last outpost that will forever resolve this. And when I reflect on Niculescu's levels of reality, and I think about that, is that a possible task? Is that something we should be pursuing, or is this sheer human arrogance? Your thoughts on that?
Oh, I have many thoughts on that, and you are absolutely right. If I came across as being very sure of myself, I'm sure of myself in my last bit here. With Rosen's story, I mean, what I'm trying to give you is that there is a way of discussing systems. But of course, when we work with real systems, then the complexity of that impredicative loop causes all the problems, because there are unintended consequences around every corner. So any complex system, if you want to try and change it, know that you're going to find that it has unintended consequences. And so that level of uncertainty about, you cannot predict the future of a complex system. So I particularly like Dave Snowden's way of thinking about, you know, what is simple, what is complicated, and what is complex, in terms of how you go about handling that. And so with a complex system, you don't understand the causality inside the system. So the best is to get a lot of experts together and talk about it. So transdisciplinarity, Basarab Niculescu stuff, you know, that's very important there, that you have a way of handling, also with methodology, how to get all of the information together. And then if you do something very small. The idea of a safe-to-fail experiment. Because if you do big experiments with complex systems, you are bound to create an unintended consequence that you didn't want. So a safe-to-fail experiment is a small experiment that you do in order to see whether it will work. If it works, you want to have a mechanism in place to amplify it. And if it doesn't work, you have to have a mechanism in place to damp it, as soon as possible. But so in a sense with the complex systems idea of muddling through. You know, there's not, oh, I'm here, I understand that, that's what I'm going to do. You say, no, I'm modest about this. I'll make a small change. If it works, okay, then I'll go that direction. So you're muddling through the system. Donella Meadows has this beautiful image of you dance with the system, with a complex system. But as you sit there, there's amazing few unintended consequences about anything that happens in you. Okay, you're going to die at the end, that is programmed into you. But actually, as a complex system, we work quite well. But it's a millennia of evolutionary processes that has designed you as you sit there to be able to be such a persistent and stable system. But human systems, I know. Anybody who's managed systems, no, herding cats.
If I could have a follow-on question, Adi, if you'll allow me. When we look at this, there's a potential for inadvertent reductionism even when we're using complexity thinking. Without trying to be critical here, I would say that perhaps one of the aspects that's missing for me is the subjective aspect. So when we look at life and the whole question of consciousness, and if we look at Eastern thinking, so does consciousness precede the material or the other way around? Or is it an emerging property? I'm not entirely convinced that's an emerging property. There's an interesting debate. Your thoughts?
Can you see why I put an amoeba up as my example?
You're avoiding the real issues.
But even an amoeba, if you think about it, I mean, there's a huge debate, even at that level. Is there cognition? And there is cognition at a very primitive level. That amoeba can sense what's happening in the environment around it and responds to it. So even those words that we use for ourselves, I don't think an amoeba has consciousness, but how do I know? But I don't think so. So what you meant, you know, when you go up to those questions, then suddenly everything that I've said becomes, you know, rather redundant because we don't know. I mean, consciousness, I would say consciousness, I am also a reductionist. That's the point. That's what the point I'm trying to make. You have to be sometimes. To say that you're not a reductionist and that you never reduce is total and utter nonsense. Because when I wake up in the morning next to my wife, and she looks at the room and I look at the room, we see different things. When you make a model, you always have to leave something out. Yeah? The trick is to be honest about what you're leaving out, and most people don't even think about that. They make a model and they forget about what's happening. So you have to always think, I make a model, I draw a line. If there is something inside the system that affects something outside the system, that has to be included in the model. And you get this idea of radical openness, so that you can never really draw a boundary, yet we have to. And that is already a reductionist thing that we do. So that's why I say you have to be a reductionist, but be honest about what you're doing, and then think systemically after that. I hope that I skirted the consciousness question, I don't know.
Ladies and gentlemen, if you have any further questions for Jan Hendrik, he will be around for the whole day. He'll also be here tomorrow morning. Talk to him. He loves talking about his topic. And thank you, sir. That was a wonderful introduction to systems biology. We really enjoyed that. Thank you.
Thank
I would
you.
like to...
Baie dankie. Dankie.
Baie dankie.